to share it later. So thank you all for joining us. We're really excited to offer this informational session about the University of California, Riverside's College of Natural and Agricultural Sciences DNA Sequence to Success DNA Barcoding Challenge, which is going to be hosted virtually this year. Today we have Nhi Tran from the College of Natural and Agricultural Sciences. He works in the with the STEM program over there, and he's going to be talking a little bit more about the program, what it entails. I believe he'll talk about the requirements that you'll have if you do get accepted into it, and field any questions you might have. Like I mentioned, if you have questions that you want to ask or you feel like you have to go, please feel free to drop them in the chat and we'll make sure to get the answers to you. And with that, if it's okay with Nhi, he can take over and start talking about the project.
Sure. Can you hear me?
Yes, I can.
All right. Thank you. So apparently I actually have a green screen and my virtual backgrounds that I downloaded are all not working apparently. So yeah, we usually keep a few UCR backgrounds in the back, but no worries. I'm going to be sharing my screen for the bulk of this presentation anyway. So thank you very much for everybody for attending. And I'm going to switch it over to my screen here. We'll move this over. All right. So I presume you guys can see my screen? Okay,
yes.
Thank you very much. So thank you for everyone who is attending, and I guess whoever will be watching the recorded portion of this video. My name is Nhi Tran. I am the STEM coordinator at UC Riverside. But more importantly, I always like to tell everyone, especially you guys as my audience, that I was a community college transfer, just like you guys. So I started life off in San Diego, born and raised, and went to probably every community college there was. And I took every science class known to man, at least that's what I like to say. And I actually transferred to UCR during the winter quarter of many, many years ago. Opportunities like this were not available back then. And so I'm pretty glad that we have extended this opportunity. And this is actually our not our ninth year, it's actually our 8th year, but our 9th time hosting this particular research project. And it's aimed only at community college transfer students. So first our first class had maybe about 24 students, only from one community college, local to Riverside City College, and we've since expanded it to multiple community colleges. And we are taking, this year, I'm not sure how many we're taking because it's all virtual, so we may be able to expand our capacity, but we're looking at beyond 72 or more groups, so or more students. So looking forward to seeing everybody. So I'm going to give you a really pretty thorough background in terms of what is DNA barcoding, how you're going to learn virtually for this project, what it entails over this time period, and what we're going to do at the very end. So just to get started, there we go. So our project overview for a basic understanding of what DNA barcoding is. You guys have obviously heard of what a barcode is, and that's that little tiny scanner that you'll see in the back of virtually every product. It's pretty universal. And when it comes to barcoding for DNA, that's specific to identifying a species. So if you were to have a sample of virtually any organism, it's hard to identify, well, what species does it belong to? This is actually the process of trying to identify a specific species coming from genetic material. So we normally start off with a sample. Every sample, as you know, we all have DNA, and then eventually we will determine based off of barcoding what that species is. More specifically, what this project aims to do is going to be using a specific model and sample. So one of the things that I like to try to explain to get everyone to understand, well, what's the importance of DNA barcoding? Why would we want to do it? What are some applications? And that is in the environment or food. So in particular, if you're to eat, you go out and you purchase different types of food, meat, fish, poultry, whatever, how do you know you are getting exactly what you are requesting or you're purchasing, or if you go out to a restaurant? And this is actually one of those methods. So we would start off with a sample. You would extract the DNA, and normally it's a very tiny piece of DNA. But within that DNA, we usually find specific genetic markers that will be specific just to that species. And so it has to be amplified. And then we're going to sequence it. And I'll get into a little bit more details. And lastly, this is a very huge field, is going to be, well, what do you do with that sequence? Now that you got it, that goes into the field of bioinformatics. So you actually plug it into a database on computer, and you'll get some information that will help you identify specific species. So going on to the project, as we call it, is the Sequence to Success DNA Barcoding Challenge. And specifically what you will learn is DNA extraction. You'll learn PCR, polymerase chain reaction. You guys may have heard of this, or you may be learning this in your general biology labs. You may actually be hearing about this in the news. Apparently, when it comes to COVID-19 testing, there are multiple methods and ways of how you can test for COVID-19, and the PCR method is actually one of the more accurate ones. It also takes a little bit longer. You have to physically put it in a machine. It takes a little bit of a turnaround time. So there's pros and cons to using different methods. Gel electrophoresis, this is something that you may have even done in high school. It's a kind of still a standard procedure when you are analyzing this type of data. And lastly, phylogenetic analysis. And what we're going to utilize is something called the DNA Subway tool. It's actually an online freeware. And I'll give you a picture of what that site looks like, where you actually get to plug in genetic sequences, and you'll be able to pump out various different species that your sample might be able to utilize. And so when I keep on saying samples, this is what we have actually come across and chosen as our sample of choice, and that is fish. So as most people are aware, fish is a huge, and seafood in general, is a very, very huge commodity and food source for the entire world. But how do you know what type of food source you are getting? So when you go to that sushi restaurant, or you go to the market and you purchase something, how do you know you are getting what's actually labeled on there? And this is kind of a sample real world application that we've decided to choose in terms of allowing you as students to analyze and figure out, well, what do we really have? So in the past, what we actually did was ask and have you as students bring in an actual sample of fish, and we would analyze. You get to learn to analyze your actual sample from wherever you got it. Now, this particular project is going to be 100% virtual. So instead, what's going to happen is you're going to learn all the techniques from A to Z, how we would have done this in lab. We're going to give you the actual data, and we have a bunch of sequences that have already been done from other experiments. But our professors who will be going over this experiment have a bunch of unknowns too. So we kind of want to make it a little bit more of a challenge. We're not just going to give you sequences that we already know exactly what it is. We're going to give you some unknowns too. So you'll be able to work on that as a group. So I wanted to tie this down also to some real world applications. There was a couple different studies where this exact technique was utilized to identify fish. In particular, Oceana, one of the largest international ocean conservation orgs, they went out and collected samples from all over the US. They went to over 675 restaurants and markets to collect various samples from 21 different states. And what they end up finding here, you can see it labeled by state. An average of 33% of the samples they received were in fact mislabeled. So as you can see, this type of technique is utilized for real world applications. And these are things that you can even think about and expand. What other things, if this is just fish, what else can be mislabeled? Another study that was actually done was from UCLA back in from 2012 to 2015. They went to 26 local sushi restaurants and they found that over 47% of the samples were mislabeled. You also notice that some of the more expensive fishes were in fact commonly mislabeled, except for bluefin tuna. Tuna, for the most part, it's very rarely mislabeled because it's almost unmistakable. So, however, if you look at the last line here, it does says halibut and red snapper samples were mislabeled 100% of the time. So if you were to walk away just knowing one thing, that is, do not order the halibut or the red snapper if you ever go out, because you're probably not getting what you're asking for. I also want to know who got to go to all those sushi restaurants. So this is the type of research I would personally love to do. So very brief breakdown of how this would work in a real lab is typically you have an actual sample. it gets acquired. And honestly, we don't need much of a sample. Some people have brought in like a whole fish. The tiniest little sample is enough to have DNA in there. And from that little piece of DNA, you would actually extract it from that tissue. And after extracting it, you have to purify it. After purifying, you have to amplify it. So once we get like our target DNA, we want to be able to increase that number to visualize it. So that's where the PCR machine comes into play. And then after that, we have to visualize it. So it's still just a bunch of liquid. And there's a couple different methods of visualization. However, analyzing it on gel electrophoresis is extremely common. And so we would actually run the gel, you'd get an image, and you put it on a UV machine. Our machine, we just slap it on and it takes an automatic picture for us, or it even has a location where you could put your smartphone and just take a quick picture. And then from that sample, what we actually end up doing sometimes is we will ship it out to a sequencing facility to get the actual DNA sequence of our target. We actually have a facility here on UCR's campus, but sometimes we even ship it out to other organizations. Sometimes we actually ship it to UCSD. They have another genomics lab that will run this for us too. So it just depends on what's quicker at the time that we need to do it, and cost is a consideration. And here, after you get the data for the sequence, you run it through a database. And the database we're going to be using is called the DNA Subway, which is kind of how it's been incorporated into the name of our project. And the DNA Subway is a freeware. This is something that you could just Google right now, go onto the website, and anybody can use it. It's basically a database that has tons of samples of various sequences from various species or lines, as you can see, of different types of organisms. So I think one year we actually did the plant line, and we had students bring in samples of different tea trees, salvia trees from the local area. But we decided that fish was a little more interesting. And on top of that, they had a really great database. So they actually have a fish line too. I think when I first started, there was only 3 lines. And as you can see, it's been expanded to 5 total. So this is a very, very great tool that anybody could utilize. And this is how you're going to get to that sequence and plug it in. Okay, so general overview of the project. We've actually decreased the amount of time this project's going to be hosted. In the past, we've had it for 4 weeks, but for the sake of being virtual and timing, we've reduced it to 3 weeks. And so within these 3 weeks, you only need to attend 2 actual days. And of these 2 days, it's very limited. You don't have to commit too much time per se. The first day is going to be Saturday, October 24th. So that's going to be the following Saturday. And we've actually had the time listed as 8:30 to 12. I will tell you guys right now that we're aiming to start at 9. So as those who log in early, we'll probably just do a few icebreakers and get to know everybody. But we're going to start at 9 o'clock sharp. And we're also aiming to end before 12 o'clock. So I know you guys are probably looking and saying, ooh, 3.5 hours, that's a long time. I'll tell you right now, we're not trying to keep you for the entire 3.5 hours, but that's the time that we've allotted. During that day, you are actually going to be watching a live demonstration. So our professors are actually going to be in the labs at UCR, and these are the exact same labs that you would have gone into if we hosted this on campus. And they're going to run through the experiment. They're going to give you a little bit of a background for molecular biology. They're going to show you the actual materials, and they're going to run it live right in front of you just to show you exactly what would be done and how you do it, and so you could see how everything works. And the great thing is you get to do this all in the comfort of your own home. So you don't have to drive all the way to UCR at 8 in the morning and find parking. Parking is usually not an issue on Saturday, and we pay for that anyway. But you'll be able to learn this in a live setting. So it's going to be a live lab class. Now, immediately, or not immediately, but later on in this day, we're also going to host a debriefing session. So understand that this is more like a lecture. And it's a lecture where not everyone's going to understand everything immediately. So myself, as well as some of our peer mentors, we're going to make ourselves available a little bit later on in the day to provide a debriefing. So basically a discussion session where we'll be able to break it down and go over these details again, just for you guys, so you understand what you just learned and what is expected over the next 3 weeks. So the next 3 weeks, what happens is you're going to work in a group, and your groups will be within your community college. And the goal is that you will be presenting on day 2, which is right here, November 14th, from 9 to 2:30. We're going to host a actual symposium. So it will be a research symposium style presentation. And you as a group will, during those 3 weeks, have analyzed the data that we've given you. You're going to write a formal abstract, and you're going to get critique on the abstract from one of our faculty. And then secondly, you're going to put together a PowerPoint presentation as a group and present it as a group. It's going to be probably 10 minutes per group in terms of presentation. And we're also going to give you an additional question. We want you guys to answer or explore on your own what are some real world applications you think these techniques can be utilized on. So in addition to talking about what you did, how you did it, we want you to do your own work, and we'd be very interested to hear what you have to say. Okay, so this is the slide that we have. If you were coming to UCR, I actually had one of my former students make this, and it's just good to give you a visual of what we would do. You know, you would just get basically a dime-sized piece of fish, bring it in, have a sample, label it. That's like the most important thing, is to put all this info on there. Obviously, you will not be doing this. We do not want any fish from any of you guys, but just so you know, this is how we would run it if you were physically on campus. And this is not to say that you wouldn't potentially do this sometime in the future. All right, and here are some pictures that I'm gonna go way back in history. This is actually our very, very first class ever. The first time we ever did Sequence to Success was back in 2013. This entire class is comprised of Riverside City College students. And they've all have since gone on to bigger, better things and been involved. A lot of them have transferred to either UCR or many other UCs, and they've gone on to all of their professional goals. Here's another look at the class. In particular, right here, she's one of our favorite success stories, is Claudia Marin. She actually started at RCC 2013, came in as a transfer, graduated in 2 years. She came in through one of our research programs, and she actually did her PhD at UCR in environmental sciences. And in fact, she's already done with her PhD by now, and she's an adjunct professor already at Riverside City College. So that was her goal, and I'm pretty sure she's well on her way to becoming a tenure track professor there at some point. So yeah, and this is just starting from 2013. 2015, class grew a little bit bigger. I think by then we actually had the UCR sign. They finally installed that up. And this is one of the other things we are known for, is to feed you very well. Sorry, you're gonna have to imagine all the food that we would have given you. But it's okay, you get to save a few pounds. 2016, we did it again, and this particular picture is very important to us, because right here in the middle, we actually have, well, we have a couple of professors. This is Dr. Jim Burnett. He's going to be leading this particular lab with a couple other colleagues, Alex and his wife. And then Darcy here, she was a UCR alum. She got her PhD and she was a postdoc at the time. And now she's actually a faculty at El Camino. And guess what? She's a professor of biology, and now she sends her students to do this program as well. And right here in the middle is actually Ms. Rochelle Campbell. I forgot to put a camera here, but if you guys have ever heard of the last name Campbell, or if it sounds familiar, and that's because she is part of what we call the Campbell biology book. If you guys have actually taken that back in bio, or back in high school, or even currently for general biology, the Campbell biology textbook was written by Dr. Neil Campbell. He was a UCR alum as well. But what's really interesting with Dr. Campbell was that he was an adjunct, or he was a faculty at San Bernardino Valley College. He taught at a community college. So they are huge benefactors and proponents of helping community college students. Unfortunately, Dr. Campbell had passed away back in 2004. However, his widow, Rochelle, is a huge proponent and benefactor for helping improve science, as well as particular for community college students. So she got to attend that particular time. And the lab that we run all these experiments out of is actually called the Campbell Science Learning Laboratory. So it's actually Campbell Hall. The entire building is named after them. These are some of the fish samples that were brought in. As you can see, that's all we need, just a tiny little piece. And some people even bring like a whole little tiny fish. This was back in winter of 17. We did an additional experiment. And so the experiment you're going to see is actually run just right out of here. And we normally have you guys do posters, and we do an actual symposium on our campus. Here's actually Dr. Scott Herrick. He is our biology professor over at RCC. Some of you have probably taken him. And yeah, we've had many other faculty from other community colleges come and support you guys as students. So we'll be inviting any and everybody to this virtual symposium, just so you know. And here's a picture of the winter symposium here. Again, as a poster presentation, this is one of the groups. Right here is actually John Palacios. He ended up transferring to UCR and he became one of my peer mentors. So he was hired, has already graduated, and he's actually starting his PhD program at UCI right now. So he's actually in the mathematics peer math program. So again, project overview, these are the details again. 3 weeks, you're only required to attend 2 specific days. In between these days, you're going to be working in groups. We're also going to give you support. We're actually going to have peer mentors and previous transfer students or current transfer students who did this exact project over the past few years. They're going to be assigned to you and provide you support. We would actually expect you guys to meet up minimum 2 times per week virtually, as well as probably create like some Google Docs and really communicate and connect online. And again, the goal is going to be to have a presentation 3 weeks later, which you guys will all be able to do, and we'll be organizing on the back end. And it'll be a great presentation. All right. So that's pretty much the end of my story. Thank you very much. This looks very strange with my green screen here, but I would like to open up the floor to any questions.
Yes, please feel free to unmute yourself and ask any questions you might have, or if you are more comfortable, you can write it in the chat and we can answer for you.
All right, and good to virtually see you, Beta. So Beta is a biology faculty over at Mount SAC. And hello, Ms. Bauchi. She is the new STEM coordinator faculty over at Mount SAC as well. Hey. Okay. Hello. And then I know we have a few students. Do you guys have any questions for me? Even as faculty, I could let you know more about the program, how this works.
Yeah, I am brand new. I'm just here to learn.
Okay, no problem.
If you want to just maybe briefly talk about the application, I know it's pretty straightforward, and the requirements.
Sure. So the application we have forwarded to every single either STEM coordinator or faculty from respective campuses. So you would either be getting it from Katie or Patricia or Bauchi or Beta. If you're from RCC, you'd get it from Dr. Herrick. If you're from Crafton, you would get it from Dr. Jonathan Townsend. I think that's mainly it. I don't think El Camino is in here, but Dr. Darcy McClelland would actually, or Descalzo would be given that to you if you're from El Camino. Application is pretty straightforward. It's a Google Doc or a Google form. Asks a lot of demographic information, a little bit of your academic history. And at the very end, most of it, you're gonna be able to plug it in. It should probably take about 5 minutes for the first part. And then at the very end, we have a couple questions. And it's just asking what type of research interest you have, what are your educational goals, what are your academic goals, and how do you think this particular experience is gonna help you achieve those goals? It's really the only part where you're gonna write something. Everything else is gonna be pretty simple. So I honestly don't think it takes more than 15 minutes to complete, but you might want to, if you want to take time to go all the way to the end to see those questions again, you're perfectly welcome to do so. Our deadlines, we initially try to have a priority deadline by this Sunday. Again, keeping in mind that it doesn't take too long to apply, but just so you know, we're going to leave the application open. If you end up applying a little bit later, we'll still try to consider, but the earlier the better. So I would encourage everyone to apply as soon as possible. This is open to everybody. Really, we're looking for science majors. As you can tell, this is going to be a mainly life sciences, molecular biology project, but anybody is open to anybody. I know we've in the past had students from all types of disciplines. I've had tons of engineering students, physical sciences, life sciences. You don't have to have research experience. It's perfectly fine. We do prefer that you've had some type of laboratory experience. If this is your very, very first quarter at community college, might not have enough science, I'll tell you that, or a little bit of background to kind of get a grasp of the understanding. And on top of that, we usually do this every single year. So you would technically have the opportunity to do this again next year. but students who have actually at least taken some courses, if you're ready to transfer, if you already have most of your classes taken, then you're probably going to be given a little more priority than those that just barely started this particular quarter. And yeah, let's see. Well, we have different funding sources, and now you don't have to be a U.S. citizen or permanent resident. You could be undocumented or DACA. It's perfectly fine. In the past, we've had different funding sources, and some of them were from federal granting agencies. And so some of those agencies did have requirements, but for this one, it's virtual. There's actually not as many costs, I'll tell you that. So we're trying to expand this to as many people as we can. So any and everybody, you are encouraged and welcome to apply. Science majors primarily. You guys are all at community college, so you already hit that particular requirement or that eligibility.
I had a question. My students, by the way, loved this last year. So our students, Bouchies and mine, a bunch of them wanted to transfer to UCR, a bunch of them did transfer to UCR, and a lot of them really liked interacting with campus, which is very difficult to do, but the students on campus, the grad students who help with the activity, the transfer students, the former students in the program, is there any chance that they could do like a Zoom and a Q&A of students while this morning session goes on so they could hear a little bit about campus, or is it just too streamlined?
I'm actually extremely glad that you brought that up. So we had a discussion about what we were going to do and how we're going to do it in terms of provide support. So typically I have a peer mentor team that I have to train in order to provide the support. But we only have so many students, and what we decided was we actually would love to reach out to both the alumni who may have come to UCR. So we obviously are connected, and we know all the students who end up transferring who did DNA Subway in the past. But on your respective campuses, that's one thing that we also thought of was, well, what if you still have students who did this last year and they're still at your community college and they're interested in doing it again? To be honest, it's it's probably not going to be too beneficial. They would do the same thing, and we also understand, like, they want to get that opportunity. What we would ask and encourage is that if you have students who did this last year and are interested in participating and giving back, we'd be more than happy to actually help train them to mentor your current group of students that gets accepted. So Patricia, Beta, I know if you have other students, I know Patricia, you have tons of students. Yeah, if they're, I'm pretty sure they're still connected, and if they wanted to help out a current group of Norco students who are participating in DNA Subway, what I can do is provide them professional development. So what I do is I actually will train them and give them a little more background, as opposed to just kind of be like, hey, take care of it. They would actually be invited to the meetings that I would host. And that's the other extra thing, because I happen to have a molecular biology background. Then I would explain to them, okay, this is how DNA works. This is how, the PCR, and this is why we're doing what we're doing. And also really just to break it down for the students. So they're gonna kind of act as they're gonna be peer mentors. They're gonna be essentially almost like TAs to an extent of helping that particular group. So by all means, and of course, I'm sure you have all their names, Beta, so do I, but you would have more contact with them. What we plan to do here is we are selectively targeting the students that we've been connected with who have already successfully transferred, and they're also going to act as peer mentors or assistants in this league too. So students who were from Norco, those that end up coming to UCR and became part of our programs, then we still are connected. I'm going to reach out to them anyway and see who would like to volunteer. But if you have them on your campus, by all means, more than welcome.